Hi guys, this is the third biochemistry lecture. Our topic today is TCA cycle. After getting our end product of the glycolysis, which was pyruvate, this pyruvate is going to go through one of the three reactions. First, oxidative decarboxylation to acetyl-CoA. Oxidative decarboxylation means first oxidation when we lose hydrogen. Decarboxylation, removal of a carbon atom from that pyruvate. Remember, pyruvate is made of 3 carbon atoms, which means removal of one of them to produce acetyl-CoA. So acetyl-CoA is made of 2 carbon atoms. Why pyruvate is made of 3 carbon atoms? Because glucose was already made of 6 carbon atoms. During the cleavage of fructose 1,6-bisphosphate, we had two products. Each one of them consists of 3 carbon atoms. Until the end of glycolysis, no change occurred in the number of carbon atoms. That's why pyruvate is made of 3 carbon atoms. The oxidative decarboxylation of pyruvate to acetyl-CoA is catalyzed by an enzyme known as pyruvate dehydrogenase complex. This reaction is irreversible. This complex has highly oxidized capacity in the cardiac muscle. Cardiac muscle is always pumping. From the first month during pregnancy till death, so it requires such an enzyme which has highly oxidized capacity to be always working without a lag. Then, the second alternative fate would be the carboxylation of pyruvate to oxaloacetate. Carboxylation is the opposite to decarboxylation. So here we are going to add a carbon atom to pyruvate to give me a 4 carbon atom product, which is oxaloacetate. This reaction is catalyzed by pyruvate carboxylase enzyme, and it's irreversible. This enzyme actually requires biotin, and that's why we call this reaction biotin-dependent reaction. The oxaloacetate, which I have produced here, will be used in either the TCA cycle or gluconeogenesis. We are going to go through gluconeogenesis in the next lecture. Finally, pyruvate may be reduced into ethanol. As we mentioned before, reduction means acception of hydrogen. This reaction occurs in 2 steps. And the first step is the decarboxylation of pyruvate to something known as acetaldehyde. This reaction is catalyzed by thiamine requiring pyruvate decarboxylase. Thiamine requirement because it requires thiamine. Decarboxylase means it removes carbon atom. Then that product will be converted to ethanol. But remember, the reduction of pyruvate to ethanol does not take place actually in human. It just occurs in yeast and other microorganisms. And let's recap this part. First, the oxidative decarboxylation of pyruvate gives me acetyl-CoA, which would enter the TCA cycle. Carboxylation of pyruvate to oxaloacetate. This oxaloacetate would be used either for the TCA cycle or gluconeogenesis. And finally, the reduction of pyruvate to ethanol, which does not take place in human body. Here's an overview for the cycle. The cycle is known as tricarboxylic acid cycle, actually because it contains 3 acids as intermediates, or known as citric acid cycle or Krebs cycle. This cycle is oxidative catabolism of the carbohydrates, amino acids, and fatty acids, which would be finally converted to energy and carbon dioxide. energy as ATP. Carbon dioxide is waste product. This cycle occurs totally in the mitochondria in order to be close enough to the ATC. ATC actually is located in the inner mitochondrial membrane, so that NADH and FADH, which would be produced by the cycle, would enter the ATC to be oxidized. And that's why the cycle is aerobic, so it means oxygen as final electron acceptor. Catabolism of some amino acids can be anaphoretics, which means they generate some intermediates of the cycle. So I'm going to break these amino acids to get the carbon chains to be used to synthesize intermediates for the cycle. The cycle is not only for catabolism, it also provides intermediates for other anabolic reactions, such as synthesis of sugar and some amino acids, and heme. Heme is an important thing in the hemoglobin, which is the oxygen transporter in the red blood cells. Oxaloacetate is going to be condensed with acetyl group from acetyl-CoA. Remember, acetyl-CoA was produced by oxidative decarboxylation of pyruvate. Oxaloacetate would be regenerated by the end of the cycle, so it's generated again when the cycle is complete. As I said before, acetyl-CoA is made of 2 carbon atoms. So we have here 2 carbon atoms which enter the cycle as acetyl-CoA, and you are going to leave our cycle as CO2, which is a waste product. Pyruvate is not the only source for acetyl-CoA. Fatty acids are also a source. Oxidative decarboxylation of pyruvate by pyruvate dehydrogenase complex, which I have just discussed, is known as link reaction because it's linking the glycolysis with the TCA cycle. And pyruvate, which was the end product of the glycolysis, is going to be transported to cytosol into mitochondrial membrane. As we mentioned before, this step occurs in order to ensure that the cycle is close enough to the ATC for oxidation of NADH and FADH. The enzyme is known as pyruvate dehydrogenase complex, actually because of the complex is a protein aggregate of 3 enzymes, which are pyruvate dehydrogenases. These enzymes are enzyme number 1, enzyme number 2, and enzyme number 3. The complex also contains 5 coenzymes that work as a carrier or oxidants, as TPP, fat, NAD, and lipoic acid. And finally, CoA, which is not mentioned here, but add it, please. CoA is another, or is the 5th coenzyme. As I mentioned, there are 3 enzymes here. Enzyme number 1 requires TBP as coenzyme. Enzyme number 2 requires CoA and lipoic acid as coenzymes. And finally, enzyme number 3 requires fat and NAD as coenzymes. What if I have a deficiency in that enzyme? Actually, a deficiency in that enzyme is very rare. But when it occurs, it causes me something known as lactic acidosis, Why does lactic acidosis take place? I have a deficiency in the pyruvate dehydrogenase complex, so that pyruvate would not be converted into acetyl-CoA. And instead, this pyruvate would be converted to lactate. Remember, lactate is acidic, and its accumulation would create problems in my brain. It would lead to neurodegeneration and death, and of course, reduced amount of carbohydrates. Why? Because carbohydrates here cannot be metabolized, because I have a problem in the enzyme which is involved in the link reaction that catalyzes that links glycolysis and TCA. Another application for that enzyme is thiamine deficiency. As I mentioned before, thiamine is very important for the first enzyme of the three enzymes found here in the complex. Any deficiency in the thiamine causes central nervous system problems, because our brain actually is totally dependent on ATP produced by TCA cycle. Then we have something known as arsenic poisoning, which we have discussed in the previous lecture, but we are going to go through it briefly again. It inhibits enzymes which requires the lipoic acid as a coenzyme. I have just said that enzyme number 2 requires lipoic acid. So arsenic is going to inhibit dehydrogenase, which is the pyruvate dehydrogenase complex. And actually it is going to inhibit another enzyme, which is alpha glutarate dehydrogenase. You would know it during the discussion of TCA cycle. Again, this would inhibit the link reaction, and pyruvate would not be converted to acetyl-CoA, and instead it would be converted into lactate, which is going to accumulate and affect our brain. Remember, arsenate also interferes with the glycolysis, as I mentioned in the previous lecture. As it interferes at glyceraldehyde 3-phosphate step and decrease ATP production. This ring reaction, since it is irreversible, it has to be regulated. Calcium would activate the BDH phosphatase or pyruvate phosphatase. This pyruvate phosphatase is going to dephosphorylate. Dephosphorylate means removal of phosphate group from our complex, which is BDHC. This would activate the enzyme, so activating the link reaction. And instead, ATP, acetyl-CoA, and NADH would activate the BDH kinase that phosphorylates, means addition of phosphate, to E1 of our complex, leading to inactivation of the link reaction. Why does calcium activate pyruvate dehydrogenase complex? Because I require pyruvate dehydrogenase complex to work in order to produce ATP by TCA and ETC. Since the presence of calcium in the cytosol means that action potential had reached the muscle and it needs to contract now. While the presence of ATP, acetyl-CoA, and NADH mean that our cell does have enough energy. So we need to stop the link reaction in order not to enter the TCA cycle, because we no longer need any energy. Okay, now let's go for the TCA cycle. First of all, oxaloacetate would be condensed with the acetyl-CoA as we mentioned before to give me something known as citrate. This condensation is irreversible and it's catalyzed by an enzyme known as citrate synthase. Actually, once the citrate is produced, it inhibits the enzyme which actually leads to its production. So citrate would give me something known as negative feedback for citrate synthase. In addition, citrate may affect phosphofructokinase. We mentioned this step in the previous lecture. Why? High concentrations of citrates means more ATP would be produced and we no longer need glycolysis, so it has to be stopped. Citrate activate acetyl-CoA carboxylase. What does this mean? Actually, acetyl carboxylase is very important for the conversion of acetyl-CoA to fatty acids. So, once I activate this enzyme, this would limit fatty acid synthesis. The next step is the isomerization of citrate to isocitrate, which is catalyzed by an enzyme known as aconitase enzyme. It's inhibited by something known as fluoroacetate. This fluoroacetate actually is a toxin which is derived from plants and may be used as a pesticide. Then isocitrate would be converted to alpha ketoglutarate by irreversible oxidative decarboxylation. Oxidative decarboxylation means removal of both hydrogen and carbon. So I would have carbon dioxide as my product and NADH as product. This reaction is catalyzed by an enzyme known as Isocitrate Dehydrogenase. This enzyme is activated by ADP and calcium. Why? ADP means cell is derived from energy, so I need to activate the cycle to get ATP. Calcium means action potential has recently arrived to the muscle cell to tell it that it needs to contract. So contraction of the muscle need energy, so I need to activate the cycle. Instead, it's inhibited by ATP, meaning that we no longer need ATP synthesis, and NADH. Why? High concentrations of NADH means the NAD, which is required for accepting hydrogen, is depleted. So we can find no one to accept our hydrogen. So by depletion of NAD or by increasing NADH, our cycle is going to be stopped. Again, alpha ketoglutarate is going to be converted to something known as succinyl-CoA. This reaction is catalyzed by something known as alpha ketoglutarate dehydrogenase. This enzyme is activated by calcium. What we realize here that most of the enzymes of the cycle are activated by calcium because calcium is involved in the contraction of the muscles. It's very important. Presence of calcium means we need energy. Need energy for myosin to pull the actin, as you know in physiology. Alpha ketoglutarate is converted to saccharide-CoA by oxidative decarboxylation, means removal of hydrogen and removal of carbon. Again, we have here carbon dioxide and NADH as our products. Instead, the enzyme is inhibited by NADH as saccharide-CoA. By NADH, as I mentioned before, the hydrogen acceptor is depleted. And NADH production have exceeded the oxidative capacity of ETC. We have mentioned this part in the previous lecture. Saccharide-CoA inhibits the enzyme because this means high concentrations of acetyl-CoA would lead to high concentrations of ATP, so we no longer need ATP synthesis. This saccharide-CoA actually contains high energy thioester bond, the same that's found actually in the acetyl-CoA. This high energy thioester bond, when cleaved, the saccharide-CoA will be converted to something known as saccharinate, and this would derive the synthesis of GTP from GTP. This is known as substrate level phosphorylation. Why known as substrate phosphorylation? As we mentioned before, because the GTP or ATP synthesis is derived from phosphate group, which was given by the substrate and not ETC. Remember, here is a very important note that alpha ketoglutarate can be produced by oxidative deamination and transamination of amino acid glutamate. Remember, saccharinate may be used in heme synthesis and be converted to pyruvate in case of gluconeogenesis. Finally, after getting and reaching saccharinate, this saccharinate is going to be oxidized into fumarate. Oxidized means that it would lose hydrogen. Here, our hydrogen acceptor is fad, and so reduced fad is our product here. Here's an important note. Before going to the note, remember that this reaction is catalyzed by Citrate Dehydrogenase, which is embedded in inner mitochondrial membrane. And here's the note. This enzyme acts as complex 2 in the ATC. Fumarate is going to undergo hydration, means addition of water. This is opposite to dehydration, to give me malate. This reaction is catalyzed by fumarase. And finally, malate would be oxidized into oxaloacetate. Again, oxidized means loss of hydrogen. And our hydrogen acceptor here is NAD. And NADH would be the product. This reaction is catalyzed by malate dehydrogenase. As you see here, actually the enzyme gets its name from the reaction. Malate because it affects the malate. Dehydrogenase because it removes hydrogen. Oxaloacetate can be also produced by transamination of amino acid aspartate. As you have seen, the Krebs cycle is regulated by 3 enzymes. Citrate synthase, which actually was inhibited by the citrate, and Isocitrate Dehydrogenase, which was regulated by calcium ADP, which stimulates it, while it's inhibited by ATP and NADH, and finally alpha-ketoglutarate, which is inhibited by succinyl-CoA and NADH, while activated by calcium and ADP. The regulation is going to affect my cycle, affect ATP synthesis. Before ending the lecture, we are going to go through the energy produced by the cycle. Here, we have produced actually 3 NADH. The first NADH was produced by the conversion of isocitrate to alpha-ketoglutarate. Here is the first NADH. The second one was produced by the conversion of alpha-ketoglutarate to succinyl-CoA, and finally, the final NADH was produced by the oxidation of malate to oxaloacetate. And so we have 3 NADH. Each one of them is going to give me 3 ATP. 3 multiplied by 3, 9 ATP. One fat H was produced by the oxidation of saccharate to fumarate. By entering the ATC, this fat H will give me 2 ATP products. 1 GTP was derived by the cleavage of the high-energy thioester bond. This means the total production of ATP by TCA cycle is 12 ATP products. And here is the end of our lecture. I hope you found it this easy.